DNA barcoding method used to one of the molecular taxonomical tools. This method is very useful to identify the single gene by a given species through the comparison of nucleotide sequences. So, we are selecting the two type of the organisms. Justify how the organisms are arranged based on the DNA sequence. So, in the past, scientists had to rigorously study different species under a microscope, and they have to distinguish them by their physical features. So, however, this method proved to not always work. Think out this way. Two vastly different species can look at same physiological features under a slide in a petri dish. So, it is analysis of their DNA sequences that aid scientists in grouping different molecular diversity. Barcoding is a vital tactic used in microbiome analysis. So, you can identify a microbe based on the ribosomal RNA sequences. So, the barcode is attached to its RNA sequences under in PCR amplification. So, the adaptations are, remember that you can identify the microbes based on the ribosomal RNA sequence. So, remember that adapters are there because they are used for PCR amplification. Every 16 years sample contains an adapter that has a unique barcode attached. So, however, the adapters are not unique. So, such as 10 different samples collected, his goal to study the different genetic makeup of each sample and compare them to one another. He would difference barcode placed at different organisms on each sample. So, simply said, once barcode or touch, the samples are to go together in a laboratory. So, this is because the simultaneously RNA sequencing method is extremely cost efficient and provides data and much more efficient in the fast way, and so they study the individual samples to help make sense of all the microbial samples, so which will hopefully lead to the inferences about the microbial population. To be more specific, once all the samples are barcoded, the NGS process begins. NGS means next generation sequencing, DNA sequencing process. Semiconductor chips are essential to the ion torrent. So, the complementary moray another scientists gave that information perform massively parallel sequence CMOS chip. So, now the ion torrent provides the nucleotide sequences, but it's up to researchers to make a sense of data. Some are the genius and studying purposes. So, these programs linked to databases such as so, scan for conserved regions which are stretches highly similar sequences among the different samples, which are the stretches of highly similar sequences among the different species. So, question will be alike. So, overall barcoding is a strategic method used for scientists, the study of next generation sequencing method. So, this organization method has saved many scientists and researchers for the time and money. So, the next time do barcoding the method favors to think about the importance of barcoding in the scientific world. Thank you.